using the hashtag Kavli Convo. And for those of you who are here in person, please know that we're selling copies of Ed's fantastic book, I Contain Multitudes, New York Times bestseller. And the bookseller informs me that he will take credit cards. So that's a plus. So, and if you're very nice to Ed and don't ask him really, really mean questions, he might sign your books afterwards. So at this point, I will turn matters over to Robert Lee Holtz Jr., science writer at the Wall Street Journal, and a distinguished writer in residence here at the Carter Institute of Journalism at NYU. And oh, I forgot to introduce myself. I'm Dan Fagan, and I'm a professor of journalism here at NYU and the director of the Science, Health, and Environmental Reporting program at NYU, and also the Science Communication Workshops. And someday I'll explain what both of those things are. Lee, take it away.
So thank you. So welcome to the Kavli Conversation on Science Communication. As Dan says, this is our first of the fall series. Now, our idea here is to bring together a leading researcher and an esteemed science journalist to explore how they both best bring the general public into the community of discovery. These conversations are sponsored by the Kavli Foundation and by the NYU Science, Health, Environment Reporting Program under the able leadership of Dan Fagan. So I want to take a second just for a look ahead. On October 13th, we're going to be seeking the punchline in science with best-selling humorist Mary Roach and former NASA roboticist Randall Munroe, whom many of you may know in his guise as the cartoonist who created the webcomic, I have to get this right, XKCD. On October 25th, we'll dive into biodiversity and the end of life as we know it with Pulitzer Prize-winning New Yorker writer Elizabeth Kolbert and Duke University extinction biologist Stuart Pimm. On November 10th, we'll tap the animal mind with Barnard College psychologist and author Alexandra Horowitz, whose next book comes out by some strange coincidence shortly before our session, and science journalist Jennifer Ackerman, author of the New York Times bestseller, The Genius of Birds. And then we'll wrap up on December 1st with NYU social media scholar and author Dana Boyd and Wall Street Journal technology columnist Christopher Mims on computers, behavior, and big data. But tonight, everyone, we are going to ride the wild paradigm. So look around. You are all part of the great microbial cloud. We are now sharing, we're inhaling, we're respiring each other's microbes at the rate of, I hear 37 million at a pop, whatever. But we are becoming a great coral reef together, a colony creature. So there is this powerful new idea in science, the microbiome, and that's the influential collection of trillions of bacteria that live within us and around us and that shape our health and our well-being in ways which we are only beginning to understand and which, in turn, of course, we shape as well. Now, as we go, I encourage our audience here to interrupt us with your questions, but please use the microphone so the rest of the world can hear what you're asking. And those of you online, please tweet your questions to us using the hashtag KavliConvo. We start our conversation this evening with a guiding question. What can we learn when a leading journalist who covers microbiology and an eminent scientist who researches it and reaches out to a mass audience, what can we learn when they get together to exchange ideas? What do their differing perspectives tell us about how the news of science is changing, how it reaches the world, and how we can do it better? I mean, let's find out. So we're joined this evening by, on my immediate left, Susan Perkins, who is the curator of molecular systematics at the American Museum of Natural History here in New York. Now, she helped organize the museum's remarkable exhibition on the microbiome, which I'm sad to say is just closed. The Secret World Within Us. She is the co-author of Welcome to the Microbiome, Getting to Know the Trillions of Bacteria and Other Microbes in, on, and around you. And I should tell you that she is a game designer. And she, by day, studies malaria parasites and the microbiome. And shortly after we're finished with her here, she's leaving for a field season in Indonesia, where she's going to be tracking lizards and other creepy crawlies for the bacteria that they, and the parasites that they may contain for study. And then down at the end there is the estimable Ed Yong, an award-winning science writer at The Atlantic. Now, he blogs on the natural wonders of biology, not exactly rocket science. And as of this morning, you no doubt will be entertained to know that his TED Talk on zombie roaches and other parasite tales has been viewed 1.4 million times. And he is, of course, the author of a wonderful, witty, and well-regarded new bestseller called I Contain Multitudes. Now, I think we should begin by defining terms. The microbiome. Now, it's the source of great scientific surprises, considerable confusion as well. And if you actually Google for a definition of the microbiome, as I did this afternoon, you'll get 260,000 different answers. So let's define our term. Susan, what do we mean by the microbiome?
Well, it's not surprising that you get 260,000 different answers because it's been used for a really long time, and I would say it's used in two ways. One is the collection of organisms, and I would say that's become the prevailing definition that we use. So we talk about the human microbiome, all the microbes that live in and on our body. We can talk about the New York City sewer microbiome, all the organisms that live in the sewer. But it's also been equated with the genes themselves. So genome microbiome. So it's used interchangeably, but I would say, and can chime in here, I think it's really the collection of organisms in a given habitat.
Okay, so that's one definition, 259,000
Yep.
to go. Ed, what's yours?
No, I'm agreeing. I'm
Yeah?
going to ruin your debate format by totally agreeing with Susan. There are two contrasting definitions, but I think that is the one that actually takes historical precedent, surprisingly, and is the one that has become most commonly in use.
So, Ed, we've known about the great world of microbes for several centuries. We have had this idea before of the beneficial capacity of microbes, not just the pathogens that make us sick. So, I mean, why is this gaining such currency now, this idea of the microbiome? I mean, is this really new science, or is it just kind of a shift in our own psychology and our own view of our place in the world and the place of the world in us?
It's a bit of both. I like to think that the rise of the microbiome as an area of science is down to two clusters of reasons, one technological and one cultural. The cultural one you've hinted at. For the longest time we thought of microbes as germs, as antagonists that we needed to kill, as signs of dirt and disease. And I think we're only now starting to appreciate that that they can be beneficial, that the vast majority of them are benign, that they have been on this planet for billions of years before us, and that they have really profound and important roles in our lives. In fairness, a lot of those ideas are old. So Louis Pasteur, a man who's almost synonymous with germ theory, also felt that that people wouldn't be able to survive if they didn't have microbes inside them. And a lot of the people who were studying microbes in the context of, like, while others were looking at germs and disease and infectious disease, were looking at the positive aspects that the positive roles that they play in our lives. But I think it's only recently that has moved from the periphery and the fringes of biology and of popular culture into its center. And I think that brings us on to the technological developments. The reason we can do that now is that we have so many amazing techniques for studying the microbes in us and all around us. you used to have to grow these things in laboratories. You used to have to look at them under microscopes, culture them in flasks and beakers. And now I can take a swab of anything, my cheek, this chair. I can shatter all the microbes within it and look at their genes. And in doing that, identify who lives where. And that technique, those techniques have revolutionized our understanding of the microbial world and have really driven home just how many species and strains exist around us and as part of us, and what they also do for our lives.
Mm-hmm. Susan, do you find that there's like a key technological change that's enabled your research in this area?
Well, I mean, Ed just said it perfectly. I mean, we used to have to culture things, and so, only fewer than 1% of microbes will actually grow in a petri dish. And so we were severely limited in our knowledge of what was growing, even on the human body, a fundamental thing we're interested in. DNA sequencing technology really opened that, and then new next generation sequencing technology where we can get millions and millions of sequences from a single sample, just threw it to the moon. And now we have huge amounts of data that allow this really cool comparative opportunity to look at these problems.
So both of you are popularizers of a very powerful new way of looking at microbiology. And so what you're telling me, this isn't just branding.
Isn't just branding.
Branding,
Oh, no. I mean,
new, improved, 100% biodegradable.
it is new and improved. No, I mean, it's changed all of biology. It's changed my research, malarial parasites. I couldn't even hope to culture some of these lizard malarias that I work on. We can't, bring these lizards back. But now I can literally take a few drops of blood and I can extract RNA or DNA and poof, know all the genes that those parasites are expressing or have within their genomes. And the cost has gone down surprisingly too, right? I can do that for a few $1,000s. So no, I mean, it's revolutionized biology.
Okay, so now I'm curious, and Ed, I will ask you a variation of the same question in a moment, but now you're a scientist, you're a researcher, you of course live and die by publication, but of course the publications are not the Wall Street Journal or the New York Times, but your peer-reviewed industry. So what I wonder now is, why are you as a scientist, as a researcher, taking on the extra chore of actually reaching out to the public? I mean, this is not necessarily in your job description as a researcher. What motivates you?
It's fun.
Really?
It's really fun, yeah. I mean, that's my primary reason, I won't lie. It's, I'm a social person, I like interacting, but also, we're at a critical junction, right? I mean, I'm going to be bold and say we have a presidential candidate that doesn't believe in science, so it is our duty to educate people about the importance of science in culture. I mean, and for me, I don't directly research the human microbiome,
Right.
but it is such a fantastic conduit to get people to understand the importance of science because it directly impacts their own health. And it's especially powerful, I would say, because it's something that you have the ability to manipulate yourself. So we can't change our genomes just yet. Maybe CRISPR technology will be developed further, but changing our microbiome is something within our grasp. We can change our diets. We can not take antibiotics when we don't need them and impact our own health. And so I have just been excited by, this has been an easy road for me to
Okay,
do
so
that.
it's
Yeah.
fun. All right, I'll give you that. But you also have clearly got like a policy interest here that you want to see certain things get currency, reduced use of antibiotics, for instance, or something like that. So when you talk to the big us, okay, let's break that down. So are you trying to influence Ed? I mean, or is there someone else you have in mind? I mean, Ed, because Ed owns the megaphone of the Atlantic. Ed owns the megaphone of a very well-regarded blog. I mean, he's what's the right word? An influencer? Anyway, he repeats in a loud and colorful and literary way things that scientists tell him. So when you reach out, who are you aiming for?
I think that brings up a good point, right? I think we now have more of a continuum. In the past, you had scientists that were more cloistered away in their laboratories and only dealing with published papers, and then journalists who had to endeavor to try to read those papers and decipher it, and maybe they could get some quotes. But there's now a much more fluid continuum, and you can talk more about this, in that more scientists who are doing primary research are also helping out in the communication aspects. And so, I'm not saying I'm going to take over Ed's blog. I couldn't possibly do that, but I think hopefully we're making Ed's job easier, but in the flip, I think it's also making our jobs much more rewarding and engaging, right? It's, I'm happy when I have good results and I get something published in a high-profile journal, but I'm also happy when people are excited about the science I'm doing in general, whether it's my cousin or a stranger on Twitter.
So Ed, same question. Who is your audience?
So everyone. So I'm going to ask the answer to the question you posed earlier, like why do you do it? And
right,
I have two reasons. First, because it's fascinating, and second, because it's hyped. And I'll do both this in turn. So this is the area of science at the moment that intrigues me the most, because I think it's wonderful. It's full of very subversive stories. Like, microbes are kind of the ultimate underdogs. They've been neglected, they've been feared, and only now are we really starting to appreciate them. And the field has so many wonderful stories to tell, which is what I try to do in the book. It has people who've been working on squid and mice and wasps and mosquitoes for decades, and who have operated on the fringes and are now starting to see their work come into the limelight, are making discoveries that completely change the way we see the natural world around us. I have loved nature since I was a kid. I was, the kid who watched David Attenborough documentaries and read books and went to zoos and all that sort of stuff, and went to natural history museums. And the science of the microbiome tells me that all of the stuff that I thought I knew really well is actually just the surface. It's just the icing on this very deep cake that we're only starting to understand. It changes our ideas about what it means to be an individual, like how our biology works, every aspect of it, development, health, immunity, thought, all of these things. And it gives us this ability to look at things that were everyday and familiar to us, the creatures that we know, cows, elephants, ants, whatever, our own selves, to look at all these things in a completely new and wondrous light. And I love that. That's what got me into science in the first place, and that's still what excites me today. And writing the book was a total joy because of that, because I kept on learning not only about things that blew my mind all the time, but also got to talk to people who were passionate and all these great stories to tell. But it is also this field is growing very quickly. And there is, like Susan said, a huge amount of promise, but also, like any science that touches upon human health and that has a lot of promise, there is a lot of hype and exaggeration here too. And I went to a microbiome conference several years ago where I felt like the rhetoric around this field was very much the same as around human genomics at the time when the human genome was being sequenced. And it felt like just reliving history, the same optimism, the same hope, but the same kind of exaggerations and a bit of hubris, I think. You could just take the word, substitute the word genome for microbiome, and there you go. And as journalists, I think our role is to push back against that. And to say what is the science telling us now? Where have we got to? How far do we still have to go? And how are we going to get there? So it's a fine line, I think, for me in my reporting and in the book to strike that balance between conveying the excitement and the wonder that I feel about this complete burgeoning area of science, but also temper that with caution and humility.
Well, I want to pursue this for a second, and then I have a question for you. So just to linger on that for a second, yes, you do a marvelous job of conveying the incredible sort of excitement and kind of color and weirdness and quirkiness of this new field, this new way of kind of approaching ourselves and the world we inhabit as a kind of system creation. But so where do you hit the brakes? Where do you hit the accelerator? I mean, how do you serve as a gatekeeper here? I mean, how do you temper? How do you have the fun and the joy of your enthusiastic coverage of this emerging thing, while at the same time not being a purveyor of the hyperbole?
I mean, I think it's actually...
What's the conversation you have in your head?
Honestly, I think it requires less thought and strategy than you might think. I mean, the thought in my head with the book and through all my reporting is just, is this sound? Like if I see a new paper, if I see a conference talk, all of these things. You know, your first instinct as a journalist is, okay, is this bullshit? And if it's
well,
no,
if it
then
involves the microbiome, it probably does involve.
Some shit.
very good.
Yeah.
That's pun one for a very long evening. And I'm glad I didn't make it. I'm usually the one who falls first. But no,
I
thank
know,
you
but I
very
had to
much.
get it in early because.
Phew.
Tally counters, that's number one.
Yeah.
Right. I think what's been really helpful is actually taking the time to get to know the field really well. So to go to conferences, to interview people, to talk to people about where the what stories are sound, which ones aren't. Like where is the field going, and where does it need to put the brakes on? Like just as an example, there have been so many links between the microbiome and all sorts of human health conditions. Everything from obesity to diabetes to atherosclerosis to colorectal cancer to allergy, asthma, or the whole works. Many of those studies are correlative, so they come from quite small comparisons between healthy people and sick people, and you find changes in the microbiome. Often you can't tell whether those changes have led to poor health or are a consequence of them, or whether it's both or neither. Even if there is a sort of causal influence, then the questions become, are the changes in the microbes instigating the disorder, or are they just exacerbating them? There is a huge tendency, I think, to see changes and to immediately assume that they are bad in like a very tautologous way. So you compare healthy and sick people, you see differences in the microbes of the sick people, and you say, okay, those are bad differences because they're in the sick people, and those differences make that person sick. It's all very cyclical, and there's a lot of work like that. There's equally a lot of very good solid work, and I think the more you look in this field, I've been reporting on this for 10 years, the more you see, the more you get a sense of what's solid and what's not.
Yeah. May I ask the audience to put their phasers on stun, please? Thank you. No, it happens. We don't have to worry about Ed's coverage here, but you know, a lot of journalists come to you. What, from your standpoint, as a source, as a purveyor of expertise, how well do you think journalists are doing here? I mean,
I think, you know, I probably have a daily frustration of, this science headline is so far off, and, you know, I retweet it or say something about it. I think there's a lot of pressure to get that snappy, alliterative, or witty headline that grabs attention, but sometimes really hyperbolizes the science that's behind that. And so, you know, I see from my job as a scientist to do quality science and present that, you know, working, say, with our communication staff at the museum with a really solid press release that, you know, we go back and forth and fact check and get the solid science out. But after it leaves the museum's office, you know, journalists can pick it up and go wacky. I mean, we watched it happen. We published a paper on a discovery of these malarial parasites in white-tailed deer. So, you know, this was exciting because we knew that this parasite had been here, but nobody had seen it, and then some collaborators were able to get some samples, and then we learned that it was more widely distributed, and we were very excited as parasitologists. Oh, we finally found this elusive malarial parasite of deer. And the headlines were, you know, malaria first found in mammals in North America. And it's like, No, that we knew that before. And, watch out this summer, don't go near deer. No, that's not what we're saying. And so you have to, just sit back and cringe sometimes, or just watch your work slowly unfold before you. But,
So when you cringe, is you less inclined to then, be a source?
no,
I talked to this journalist for an hour, and it wasn't just that they didn't quote me, but they got the headline wrong, and now everyone in Connecticut is worried about the deers in their backyard.
Well, luckily, I hope that it becomes slightly cycled. Our work stands. It's a peer-reviewed paper. If you took the time and energy to go and read the paper, I think most people would have those myths dispelled. And otherwise, the people that are, freaking out, that's going to die away relatively quickly. Otherwise, they contact us and we try to straighten it out. But honestly, that rarely happens. We live in a culture of, the sky is falling. Oh, no, sorry. Brangelina's breaking up. So.
Right, I guess it's a variation of the Warhol thing.
Yeah.
In the future, everyone's sky will fall for 15 minutes. So let me ask you both. I'm just curious now as a personal matter. So you both have immersed yourselves in a new way, in a pervasive way in the world of microbes and microbiomes. And I'm just curious how this has changed your perception of who you are, literally. Who am I sitting with?
Okay, so it looks like this is one body powered by a single genome and driven by a single mind. And as I say in the book, it's a pleasant fiction. It's a nice idea and it fits with our intuitions. And yet we see that microbes influence just about every corner of my biology. So the development from a single fertilized egg into an adult form critically depends on signals from the microbes we carry. If you look at animals that are raised in sterile environments, they have all sorts of problems to their bones, blood vessels, guts. All sorts of organ systems don't develop correctly without that microbial contact. We know that they help to digest our food. We know that they help to build and train the immune system. The immune system is typically thought of as this defense force that stops microbes from getting inside us and that detects and destroys them. And yet, we see that the microbes within us, some of them, help to build certain types of immune cells. And then they calibrate the balance of the cells so that they're reacting to infectious threats, but they're not overreacting to harmless things in the world around us like pollen or dust or allergens in our food. And then in turn, we see that the immune system helps to select for the microbes that live within us. So it determines which species and strains get to take up shop in our bodies and where they get to reside. So it's a two-way thing. The immune system is built by microbes. It controls microbes. It's more like rangers in a national park than it is this force of soldiers and police officers. And that is just an example, one of many, of a part of ourselves, immunity, that we can see in a totally different light. So I feel that my view of myself and the world around me is greatly enriched by looking at this, which is why I start the book with a visit to the zoo. I go to visit San Diego Zoo with a man called Rob Knight, who is one of the biggest names in the human microbiome field. And he's not doing any particular study. He's more so doing a feasibility thing. He's looking maybe at doing a study where he looks at the skin microbes of various zoo animals and compares them. So all we're doing at the zoo is he's just looking at which animals will tolerate what degree of swabbing. So
I was so jealous you got to do that.
I know, it was amazing. I was just walking around going, its face is too small. We pick up the porcupine, like, can you swab the porcupine? It turns out the spines aren't a problem, but it will just bite the end. So the whole point about doing that was to take something that was very familiar, so a zoo, the animals within, and show what that world looks like to someone who sees it in a completely different light, who sees the animals as ecosystems, as like sets of islands which are home to thousands of individual organisms rather than just one in isolation. So it was just it was a fun way of showing the kind of thing that goes into, that has really happened in my mind over the last 10 years, that shift in perspective.
And now, Susan, I'll ask you the version of the same question. So I know that normally you're obsessed with parasites, but has this changed how you see yourself when you look in the mirror?
It really changed. It really changed me, and I said this when I reviewed Ed's book. I could tell he was one of the kindred, right? It's like there are those of us who see ourselves as ecosystems, and then the rest of you. So, but someday you'll all come into the fold. No, it totally changed me. I don't interact with the world at all in the same way I did 5 years ago. When I shake someone's hand, I'm like, yeah, I just got some microbes. It's probably fine. Pet a dog. Yeah, okay, I got some other ones. Like you said, I'm about to go immerse myself in a complete other ecosystem, and there's no way I can experience that whole, epic adventure without thinking of microbes the whole time as well. Like, what am I picking up now? I just ate street food in Jakarta. Hopefully nothing bad.
I'm not hearing a welcoming map being
No,
put out there.
I'm happy. I embrace it. Actually, this idea that the more diverse you are, the healthier you are, has really permeated me, and so I seek out opportunities to diversify. I think, one of the things we said in the AM&H exhibit, and it's been showing up a lot of places, right? It's like we have way less diverse microbiomes because we Western people spend so much time indoors, right? We all live in New York City here, and we go from our office to our apartment on the subway or in a cab, and so we're just cut off from the microbial world. So I'm excited to go be in the middle of it, yeah.
And I'll get some questions queued up, so I don't want to, what?
Hi. So my question is, we know it's early days in learning about the microbiome and human health, but without the hype at this point, can we have some idea of what the legitimate hopes are for what knowing about the microbiome is going to do for making us healthier?
I can rephrase that question if you don't mind. So every year globally, people are now spending $4 billion on probiotics. Is that all money down the toilet?
Almost literally.
I'm going to go with her phrase. So I think there are a few short-term wins that we can do. Where is that? Hello. So I think there are a few things that we can do in the near term that will probably have an impact. So one thing I'm excited about is stratification of medicine. So trying to work out who should get which drugs. That's a very simple thing we can do. Everything from like paracetamol, which I think you guys call acetaminophen, to the big new cancer immunotherapy drugs, things like checkpoint inhibitors that unleash the immune system in tumors. All of these things seem to vary in effectiveness depending on which microbes you have. So the microbes might break down the drug into something useless. It might change the drug into something toxic. All of these things can happen, and I think a very easy thing to do would be to look at someone's microbiome and say, okay, you should get this drug and you should not. We might also be able to drug the microbiome. So rather than change the species that live within you, trying to nudge them in a different direction. So one of the labs I think does a really good job in this field is run by a guy called Stanley Hazen. I can't remember where he works now. But they look at the links between microbiome and heart disease. Now, if you are prescribed, if you have a high risk of heart disease now, your doctor will probably prescribe you a statin, which is a drug that targets a human enzyme that affects the metabolism of cholesterol. But there are bacterial enzymes also that affect how cholesterol is, the rate at which cholesterol is broken down and deposited in your body. So if you can find chemicals, and there are some, that affect those enzymes, then you can also change the way cholesterol is laid down, and therefore maybe affect your risk of heart disease. So maybe in the future you'll get a couple of drugs like a statin to target the human half of you, and this another drug that targets the bacterial half of you. And I think maybe that's going to be a little bit, because that substance isn't an antibiotic. It's not killing any bacteria. It's just taking the ones inside you and just pushing them in a slightly different direction. Much like the pharmaceuticals that we take influence our own human biology. And I think that feels like it's going to be just conceptually and practically easier than trying to add or remove species. As we said, it's such a complex ecosystem. It is difficult to do that with any kind of precision and efficiency.
Susan,
I
please.
just wanted to chime in. I think, you were bringing this up before about the hype and trying to back away from it. And I think at this point, like we were just saying, I'm happy if the hype is just, yay, I have bacteria in me, and that's okay.
Right.
As long as if that's the hype, and if we study them more, we might get some really amazing health benefits. That's the hype I'm happy with, And what we've been trying to do, all of us here, is try to just steer it into the science that where we are now, where we know it's going. It may not be there yet, but stay tuned, and your example of the human genome really resonates, right? Because I think that disappointed a lot of people. It was hyped as, we're going to unlock the secrets, we're going to decipher the code, all these metaphors about sequencing the genome, and then we did it, and we were like, we still don't know the answers, right? We learned a lot of answers, but the microbiome has been this massively added thing of, well, we're a tiny fraction of the genes in our body, and there are all these other things. So yeah, I'm okay with that hype and at least getting people excited that the microbiome is a good thing to study.
Yeah, for sure. The idea that bacteria and other microbes are important to us and are really cool. I've got no problem. That's what what I feel as well, right? I think it's cool, it's exactly what you said. It's the idea that it will lead to massive medical breakthroughs like right now.
Right.
And I think there are also risks because so if we take something like fecal transplants, so fecal transplants are what they sound like. They're a way of you treat a problem by giving sick people stools from a healthy donor, and this has proven to be incredibly successful for treating Clostridium difficile infections, C. diff, a microbe that causes severe and recurrent and sometimes fatal cases of diarrhea. Fecal transplants have been incredible for treating C. diff. They beat antibiotics hands down. But they're also very, very easy to do, which means that a lot of people are doing them. And they're doing them for all sorts of different conditions. Because the microbiome has been linked to inflammatory bowel disease and irritable bowel syndrome and asthma.
Thinness, right?
Right, exactly.
I want to be skinny.
Yeah,
Okay.
right.
Or behavior.
Right.
Behavior thing.
Yeah.
Right.
Yeah.
Because of all of these connections, I think people are trying out a technique that seems easy. And I don't want to go harsh on that community, because a lot of them have tried this out for things like C. diff infections because they haven't been able to get legitimate help from their doctors. But this is not a procedure without risks. We know from experiments with rodents, for example, that you can transfer symptoms of a lot of different conditions, like all the ones we talked about, through moving stool from one individual to another. So it's an easy thing to do, but it's not necessarily a harmless thing to do. You do it in the medical setting. You get screening of the donor for any pathogens that they might carry. Increasingly, people are screening for all these other chronic conditions that we've linked to the microbiome. So, I think there is good and immediate reason to be cautious about how we talk about where we are with the science and what the promise is going to be in the future. I think we can talk about that.
Yeah. We have a question there. I'm sorry, you've been very patient.
Oh, no, you sort of answered it. This was a question from Dan Rubitsky, who is actually sitting right there.
Hi.
He was wondering how hard it is to kind of counteract colloquial misinformation about the microbiome.
That's a good question.
Yeah.
Yeah,
So you're the one, Ed and I are in the business of kind of creating the distortion, if you like. But you're...
Why do I have to clean up after?
You have to, exactly. You have to clean up,
No.
so you should answer that question.
I mean,
How hard is it?
It is hard. It definitely is hard. I think, you know, we've seen this whether it's with vaccines or vitamins. You know, people have very strong opinions about anything related to human health, especially if it's their own health. And so I think on an individual level, it can be really difficult to counteract people's perceptions. You know, they can say, well, no, I've done this and I've had this effect, and you cannot, and you and your data cannot tell me otherwise. You know, even if I could give them a few other explanations. But I think in a general scheme where most people are interacting with this field for the first time, whether it's an exhibit or reading a book or seeing a blog post or something, they just come in with ultimate curiosity. They keep hearing microbiome. They hear these buzzing words about fecal transplants or, you know, mind control, and so they just come in hungry for knowledge. And so practically, no, I haven't had a ton of challenges, but you are always going to have those folks who are just dead set in their opinions, and you can't.
We have another question here.
Hi, this is on the same topic in a way. I think when you talk about science, there are two things. There's, you know, information about fecal transplants or the things that live within us or the cosmos. There's also a structured form of inquiry, which is, you know, how scientists operate. And it's when you say, you know, we have a presidential candidate who doesn't believe in science, I think part of the problem with how an audience that isn't made of scientists interacts with science is they don't think about the inquiry, but about the trivia that comes out of it, or, you know, the fun facts that come out. How do you address that in your work, both of you?
So, I mean,
Yeah, I mean, I think that's hard. I mean, obviously, I was gonna say it's hard. It's hard work. I think one of the hard things for us as scientists, though, is really explaining that process of inquiry, because so many were taught that scientists operate in a way that we don't actually operate in. You know, we don't actually do the scientific method on a daily basis, right? That's rarely how things operate, especially in a high-powered publication grant-getting world. But I think the microbiome has been a really nice way to teach that lesson, which, you know, as you brought up correlations, which is the stage we're at now. We can take healthy people and sick people and see that they have different microbes, and if you have enough of those people, you start to uncover correlations. And then you start to set up experimental causation through controlled experimental design, which maybe is more in line with the traditional scientific method that you might have learned about. So, I don't know, to me, you know, I think that there's a lot of this excitement. There's the added complexity of the fact that so many of these studies are being done in non-human systems, like mice especially, and then we're being asked to translate that information to humans as though we're the same, right? And we're clearly not. But yeah, it's to me, it's an opportunity because people care about it, and there's so much cool science coming out of it, but to also use it as a very powerful demonstration of the need to carefully dial back and show determinative causes of things. So obese people might have a different microbiome, but as Ed said, did the microbiome cause it? We have to do experiments, and those take a long time. They need careful controls. Science is a slow process. That's not the stuff you want to read about. It's not the stuff you want to spend time waiting for. But that really is a fundamental truth of how this
is that the sort of narrative that you, go ahead, please.
Yeah.
So I feel like if people, I think the reason why people focus a lot on, or it seems that people focus a lot on those facts, like the end point, the discoveries, rather than the process of inquiry, is that we're not very good at talking about the latter. And by we, I mean all of us, like scientists, science communicators, science journalists. There is a thing, a concept among people who study the science of science communication called the deficit model, where we treat people as these empty vessels into which we pour information, and that's how they learn about science, and that's how we change their minds if they buy into myths and misconceptions. And it's just nonsense. That is not at all how people engage with knowledge or get engaged with the world around them. And I think the way we shift that towards appreciating science as a process, as a way of understanding the world, is by telling stories. Because we are storytelling animals. Stories create emotional connections. Why it makes people feel something about science rather than just knowing something about it. And I think that makes all the difference. And that's the approach that I've tried to go for with the book. And it's easy. The thing is it's easy. There are so many great stories in science, and not just because this field has been going along for a long time, not just because it's full of subversive characters and plot twists and and underdogs, but just because science in general is full of those stories. It's full of successes and failures and quests and passions and interests, and all of the elements that make for great narratives are there all the time. When I write a news piece about a new paper, that's the approach I'm going for, just as it is when I'm writing a 100,000-word book.
Susan, did you have something you wanted to add?
I think I was just going to say, there's so much pressure from whether it's my family or the public, is what did you find? You set up this experiment, what did you find? And the reality is I might have found something about just doing the techniques or whatnot that's going to make it better next time. So right now I didn't find out what I thought I was going to find out, but I'm in and that's not a failure. And I think, there is a lot of pressure on us to deliver, and so we're constantly doing our own narratives, whether in peer-reviewed publications, which is, well, what result did I get, and how can I portray that so it's important to other people that might be reading that?
Dan, you have a question?
I do. I'm still pondering the concept that science storytelling is easy. I'm thinking about I'm thinking about just quitting and abolishing my program, because really, who needs training? No, I totally understand what Ed is saying, that science is full of amazing stories. He's totally right about that. I'm interested in the concept of how the new knowledge of the microbiome specifically changes storytelling. I mean, besides eliminating the singular pronoun, like nice to meet you all, every time you say hello. It seems like it does more than just enhance our knowledge of reporting about infectious diseases. It's just a whole new way to think about writing about all sorts of issues that involve aggregations of people beyond just the individual human being. So I wonder, all three of you really, if you've thought about that, and what are the ways that this changes the way that we tell these stories, having this extra knowledge. And I'll go stand over here so you guys don't keep having to twist around.
The pronoun thing is a complete pain in the ass.
Yeah.
We
need a different,
Right, you guys.
So I think you're right, Rody. I think you're right, and I think that
Talk to the dark.
there are so many. There are lots of interesting conceptual things at work here. Like, how do you think about these relationships? I think for a long time, people thought of them as negative ones because bacteria were germs and whatnot. And now you've got this different narrative about good microbes and friendly bacteria. And actually, the very first thing I wrote, the very first part of the book I wrote was what is now chapter 4, in which I assert right from the start that there is no such thing as a good microbe or a bad microbe. In many ways, we are the supporting cast in their stories, and I think that's the right way to think about that world. Like, we are just another habitat in which
They
they
made
live in.
us to achieve their evil goals.
That's right. Yeah.
Yeah.
Yeah. And yeah, we are just another world in which they live in,
Right.
in which they live. We are.
But to stick with Dan's point, because he's asked a really good question here, and I'm not going to let myself off the hook on this one either. But we're not writing for the microbes, and the nature of narrative is to sort of focus, as is the nature of the research about which we write. It's one bug that does this, or one stool sample of a sort, and we're bringing this narrow casting focus, is the nature of most journalistic narrative, to a topic that both of you have been telling me with great eloquence all evening is not a narrowcast, it's broadband. So how do you tell a kind of classical journalistic magazine feature, PBS, heroic scientist doing great things to her topic here? I mean, how do you do that? I mean, I don't know the answer to that question.
I mean, I think some of it goes back to what I said earlier, which is it's a different way of storytelling because it directly connects with every single person. We all have a
Right.
microbiome, and like I said, we all have the capability to alter it in some way, maybe minor, maybe much more substantial. And that's different than our genome. It's different than other health and science stories that we might have connected with. I mean, we get excited about stories of space or new discoveries
Right.
of species, but they're not directly relatable to us. We enjoy them. There's something that piques our curiosity. But with the microbiome, we individually can do something differently to affect it, right? And I think that's really exciting to a lot of people. And if I could throw this out there
No,
as a
please.
tangent a little bit, one of the things that was exciting as well with the microbiome, one thing that was sort of unexpected with the exhibit that we discovered is how much kids immediately identify with having a microbiome. And we go into the exhibit and we say, well, most young people they can't grasp the concept of bacteria. It's too ethereal to them. They can't see it. They don't understand what a bacterium is. And then I have the experience of getting on the elevator to go to my exhibit and a 4-year-old pushing the button because he can't wait to go learn more about his microbes. And the mom saying this is the 4th time he's been, and he's so excited about taking care of his microbes. And, I mean, he might not completely grasp, his gram-negative bacteria and his gram-positive and his firmicutes and his Bacteroidetes, but he immediately grasps that he's an ecosystem, and even as a 4-year-old can do something to change that. He's eating his broccoli for a change, and it just connects to every single one of us. So.
And I think that the principles of storytelling about the microbiome are just the same as storytelling about any other aspect of science. Like, for a start, there's all the people. There are so many people who long time working in this field and piecing together amazing things. Like, I feel like you get people to care about microbes by getting people to care about the people who care about microbes.
Yeah, how do you meet Colleen Cavanaugh and not care about microbes?
Exactly,
It's
right?
infectious.
Jack Gilbert,
Jack
Margaret
Gilbert.
Fong.
Yeah.
Actually,
Yeah.
when I came up with the idea for this, the first night when I thought, this is the book I'm going to write, I wrote a 2-side Word document that had like a little essay explaining a lot of like the conceptual stuff we've been talking about, and then like a list of names. And it was all those names, like the people who I knew worked in this field and who had great stories to tell and whose stories I was going to tell. Yeah. And they are the characters in the book. But also like the protagonists and the narratives in this field aren't just them, it's also the organisms themselves. It's the field as a whole, how it has grown over time, how it has changed and expanded, absorbed all these new technologies, these cultural shifts, how different lines or how many different fields, so people who work on the human microbiome and insect microbiomes and on deep sea microbiomes, all work separately and then converged into this giant super field that we have now. So, you're not short of protagonists, some of which are humans, some of which live in humans. It's so many stories right for the telling.
Yeah.
So what kind of story can a museum exhibit tell? what kind of narrative can you construct, did you construct,
Yeah.
with the storytelling device of the museum exhibit? I mean, we tend to talk about words and writing because that's what Ed and I do, but actually there are other ways of telling stories.
Yeah.
And
I mean,
at no time in the history of anything have we had so many of those ways at our fingertips.
right. No, I mean, and we had this amazing opportunity to have visual experiences and multimedia experiences. So, to surround visitors with the microbiome. And so, in some ways it's funny, as the exhibit was closing, I pulled out our very first blueprints that we ever drew up. And that narrative was pretty much in place from the very beginning. We knew we had to get people comfortable with the fact that they had microbes and be specific about, how they are transferred. So the story of skin plays in there and where they came from. So microbes in the reproductive system. And of course we had to talk about all of these things that they're hearing about with digestion, whether it's probiotics, fecal transplants, etc. We knew we wanted to have these exciting characters. So many of the people that are in Ed's book were also in one of our videos in the exhibit because they are such amazing personalities. You connect, you see their excitement, you see their promise, but you also see their realism. And I think that was an important narrative that we tried to do. And then all along the way, it was all about you. I mean, the exhibit was the secret world inside you, and we used mirrors and we used cutouts where you imagined yourself in it because we were trying to do exactly that premise, which I mentioned before, which is every single person can connect to this exhibit. Every single one of us has microbes and will identify with some part of it, whether you were born by C-section or weren't breastfed or have an elderly aunt who had C. diff. Every one of us had connections throughout that exhibit, and so we used that inward reflection as our narrative. I think it worked.
Question here, please.
Now that we have this knowledge that we're an ecosystem and not just an individual, how easy is it to actually alter the microbiome?
So I feel it's really hard, and that's why I've been stressing a lot in the book and in interviews that this manipulating the microbiome is a much trickier thing than I think a lot of people give it credit for. I think people are not pretending, but I think people have been lulled into the sense that the arithmetic is easy. That say, you are missing a thing, much like having a vitamin deficiency and you give someone a supplement and they're better, you plug the gap, they're fine. Or you have too much of an extra thing like a virus and you get rid of that and then you're fine. But it's not, it's like these are, when I don't just say ecosystems as metaphor, they really are as complicated as a coral reef or a jungle or anything like that. So if you add species in, how are they going to compete with what's already there? How are they going to compete with each other or cooperate with each other? Do they have the right food in the environment? Do you need to provide it for them? How will they interact with the soil, say like our immune system or the cells in our own human cells? All of these questions, I think, make a huge difference, and a lot of the time they're not being considered. And I think that's why things like probiotics are very underwhelming, or at least inconsistent in terms of the evidence base around them, and why fecal transplants work really well with C. diff, but not so well, or at least not so consistently well, with a lot of other conditions. Because even when you're giving someone like an entire community of microbes, it's really hard to reset their own ecosystem. So I think we're only at the start of this, and it's there are a lot of ecologists in the field. I think this microbial ecology was a thing, but not enough. Yeah, absolutely. Yeah, I mean, microbial ecology was a thing before anyone even coined the word microbiome. And I think that's the perspective we need.
No, I mean, in the biotech companies, the last panel I did was a bunch of biotech companies who are trying to capitalize in microbiome science, and that's exactly their message is we need ecologists, we need evolutionary biologists, but mostly we need ecologists because these are the exact principles that we're dealing with. And so, but I mean, I love that question because that was something I would find myself backed into a corner on a lot of times, right? Because there is the message that we can change our microbiome, and certainly taking antibiotics when they're not necessary, especially some of the strong, I mean, you know, we all remember after 9/11, right, and the anthrax, that people were just grabbing Cipro and taking it like Pez candy, and it's like, no, this is an incredibly strong broad-spectrum antibiotic, and you are resetting a microbiome potentially to dangerous levels. So, I mean, we can change it, but, you know, on the flip side, you know, if you have a hamburger with bacon and extra cheese and double order of fries, you know, your cholesterol might spike, but you're not fundamentally going to change your microbiome. That does take more systemic long-term things, but I would constantly find myself, you know, backed into saying, well, you said you could change it, but yet our stable microbiome is very resistant to change, and which is it? And part of it is we don't really know, but I think, you know, Ed's exactly right. I mean, I loved your metaphor of thinking about it in terms of other ecosystems, because none of us would imagine that we would throw a fish species into a coral reef and suddenly everything would be totally different.
Right.
What we'd more likely expect and what happens in the microbiome is we'd come back in a month and that fish we just put in there would be gone. So,
Yep.
yeah.
So, question here?
Oh, hi. All these are very exciting and interesting conversation and content. I'm just curious, like from your perspective, what kind of microbiome research needs more public coverage, or like what kind of topics the public need to know more about microbe? It can be, I don't know, a misconception, or it can just be a cool or silly finding. Yeah.
So we'll all take a turn answering that one.
Yeah.
So I love a lot of the work going on in microbiomes in other animals. So humans are only about maybe 30%, like a quarter to a third of that book. The rest of it deals with the rest of the animal kingdom. So this is a natural history book, and that is how I wanted it to be. And I think that a lot of those other systems, a lot of those other creatures don't get a lot of play. I've been writing with them for a long time, and I think they have valuable lessons to tell us. You know, the colonization of squid by light-producing bacteria says a lot about how microbes influence our own development, and about how animals select for the right microbes, specific partners out of this huge ensemble in the environment. And speaking of the environment, I think there's a lot of work on environmental microbiomes, on microbiomes of acidic mines and oil spills and, you know, rivers, and that basic biology doesn't get a lot of play, and I think it's really fascinating. You know, there are vast swathes of microbial diversity that we did not know about even a few months ago. If you look at the family tree of all life on Earth, it looks radically different now than it did even this time last year. And I think that is amazing to me, and it doesn't get a lot of attention. And like I enjoy the chance to.
Susan?
It's funny when you asked that question, I said, well, first I'm going to answer other animals and comparative. But if Ed says that, I'm going to say environmental systems. So he's involved in this thing. So I'm left with, yes, everything he said. But then I would add, you know, and I think we're biased too, being in New York City. But I think I'm really excited about urban microbiology.
Yes.
You know, maybe I just stole
Totally
your
agree.
answer. Now
That's
we're
my
not.
answer. This is how it works. This is how it works. That's
And
my
took
answer.
both of mine. So I
Yeah.
think you know we're becoming a more urbanized society. We had a really cool, the New York Academy of Science sponsored a symposium about a year and a half ago on urban microbiology, and it's really exciting because it's a totally different ecosystem. You know, there's soils and there's coral reefs, but what about concrete and subways and high-rise towers and you know, building?
Yeah,
these
for the
new
first time in
areas.
human history, more than half of humanity, a species that evolved as a tribal, nomadic, rural, agrarian creature, half of humanity lives in a city. The microbiology of the built environment, I think, is the most exciting, most troubling, and most undercovered and least understood aspect. You want to change your microbiome, open the window. Change the air conditioning filter. The materials that we surround ourselves with, the buildings that we make, all have a very direct and perhaps profound but little understood effect on the microbiome. So.
What I love about this is that the microbiome is a deeply connective field.
Yeah.
You know, and by that I mean, we often think of ourselves as conceptually separate from the rest of nature. Like we have man-made and natural, urban and rural. And the microbiome tells us that these distinctions are bullshit. There is so much connectivity between all of these things. And the microbiome, the principles that govern the microbiome of the built environment are just the same as those that govern those in a child's body or like a cow's stomach, or all of these systems. It's the same ecological principles from one to the other. And I think that if you understand the book ends with a chapter on the microbiome of the built environment, because I want to take us out from just considering these worlds inside us to those that are around us, and how those two are deeply connected with each other.
Have a question?
Hello. Before I ask my question, Dan would like you to talk about your work on the urban microbiome.
Me?
Yes.
I'm about to become part of this story? That violates like the most fundamental canons of journalism.
I feel like you should ask your question. Because you have now asked questions on behalf of two dudes.
Yeah.
And I would like to hear what you had to say.
That's my job.
Yeah.
No one asked Lester Holt questions last night.
Yeah. So I guess what Dan is referring to is, I think it's fair to say that both Ed and Susan come to this topic with the sensibility of a naturalist, and I don't. So I'm very interested in the microbiome of currency, for instance, the microbiome of sewer systems, of the built environment, and in particular I was involved with a project that some researchers at Weill Cornell Medical College did, which is now being replicated, I have to say. It had been about a half dozen cities around the world where they use the techniques of metagenomics, which is kind of underlies a lot of the genomic research now, where you're able to kind of just take a sample, and without knowing what is in it when you take it, you can just chop it up and spew the DNA into a sequencer and reconstruct it computationally. Now, there's some drawbacks to that. If you haven't already sequenced the bacteria, you can't identify it, but that's a different problem. But in any event, they attacked the microbiome of the New York City subway system. And there were some things about the study that were problematic, and they were problematic in part because it was pioneering. People were still figuring out how to do DNA studies on this scale. But the thing that they found it got a lot of ink at the time, but the thing that they found most interesting, I think, was that, they found the DNA of, pastrami and pizza cheese and kimchi, it was like a map of the takeout food delivery system in New York. But they found that over half of the microbes that they were able to identify as being microbes, they couldn't tell what they were, but using the 16S, they could just tell that they were these sorts of organs. They had no idea what they were. They were unable to identify them. We live in a world we know not what of. And we're affecting it, not just internally, but externally all the time. Every time we put up a high rise, every time we dig a new tunnel, every time we change from an electric car, from a gasoline-powered car to an electric car. We're just, because we can't separate these things out. We want to talk about, I'm agreeing with Ed really, we want to talk about the microbiome, but then when we write about it, when we cover it, we break it down into these component parts. There's a skin microbiome project. There's a belly button diversity microbiome project. There's a microbiome of your inner elbow. There's the oral microbiome. It's just what we do. We take this holistic thing and we break it down into component parts that in fact are kind of meaningless. They're just our organizational conveniences. So when we talk about the human microbiome, I guess I put to all of us, where do we start and stop? And we're creatures who create our environments, and we create environments on a global scale. We're the 21st century termites. So when we try to cover this stuff, I think it is very complicated, because in some ways our traditional tools, and I agree with you, They're fabulous characters, and they're great stories to be told here. And in fact, they're wonderful characters that are microbes. You know, Wolbachia. I mean, gosh, what an interesting thing. But by focusing, we kind of run counter to the real story that we're trying to get at. I don't know. So I think that's what Dan was talking about. So, sir, you have a question?
Sure. I was going to ask about the sort of non-individualistic, non-health implications of the microbiome for the environment, and you sort of touched on that, so then I had to quickly shift my question. So I'm wondering if you can think of like political implications for the microbiome. This might be the idealistic, but any kind of political implications because of the way that they relate to borders and how like microbes move across borders, or possibly things like surveillance, the way that you can do forensics on we have like a microbial footprint now, and so can you think about political implications or political recommendations that you'd have based on the potential of the microbiome?
There are serious privacy concerns.
There are forensic applications for sure. So we, all of us, eject aura of microbes, and because our microbiomes differ so much from individual to individual, at least in small-scale studies so far, it has been possible to trace people based on the microbes that they leave behind in a space. You know, now whether that scales up into like
everyone's looking around.
That's right. Yeah. I think this appeared on CSI Miami. No, really. And so whether this, I think, scales up to, you know, to a microbiome database, I'm not sure. I feel like it's so dynamic, and there's so much noise in that system that it's hard for me to think about how it might produce a test that is sensitive and specific enough to be used forensically. And then
yet,
I think
yet there's a company making money selling a spray that you spray behind you to kill the microbes.
Really?
There we go.
Yes.
There we go.
Wow.
Kills the DNA of them, which, you know.
Is it bleach?
It's gotta be just bleach. Yeah.
I mean, it's like a hose with some bleach.
Yes. It's a little, you know, a little squirt bottle of 10% bleach. So you're just.
Well, bleach is effective, but many hand sanitizers. One of the funny uncovered aspects of this subway study was that they did sample surfaces before and after using hand sanitizers and discovered that, well, there was just as many microbes before as after. That these were not necessarily particularly effective things. So we have a question here?
I have a question a bit about the built environment that you were talking about, the man-made versus the natural. And it made me think about steel and concrete, and what you seem to be talking about are the surfaces of those materials, not the materials themselves. Is that right?
Sometimes, yeah.
Only because so far we've only sampled the surfaces.
But
So
are you saying that
it could be the structure, it could be the things that the materials, the surfaces are made of.
but depending on which materials those are and how man-made. So then my question is, what would you say, or how would you characterize environments that are inhospitable to microbes?
Can you name an environment that's inhospitable?
That's one of my
to
questions.
I mean, I feel like.
Well, I'm asking about steel inside, or concrete, or that's what brought me to that question, but I'm imagining there must also be environments in which it is more difficult for them.
Okay, so if we talk about the built environment specifically, I think that so it's not just like the surfaces we build stuff out of, but the way we design our buildings, or like as the internet, whether we have adequate ventilation, and that changes the traffic of microbes about the place. And I feel that again, this is kind of a new area, so I don't want to like go into too much detail, but certainly all of those factors contribute. If we look like more broadly, like parts of the world that are inhospitable to, is that what you mean, or just?
No, I mean
right.
a type of environment, not a particular place.
Okay, I mean, all of these factors determine which microbes get where and how abundant they are. But there's no part of the world that they can't colonize. Like, you know, you have microbes that will eat crude oil, uranium, metal, heavy metals. They'll live in like the most acidic mines in the world. They'll live in Antarctic ice. You know, there are people at NASA whose job it is to specifically stop microbes from getting onto things that we send into space, so that we don't we don't accidentally colonize other worlds with our microbes. So like they are everywhere. Like I feel that there isn't really such a thing as an inhospitable environment for a microbe, except possibly for like the bottle of bleach.
Yes.
Yeah, yeah. I mean, I believe it's the case that in the there's a species of bacteria that was recently discovered that exists only inside NASA sterilization chambers, and I'm not making that up.
Yeah.
I'm not making that up. Susan, do you have something you want to add?
In terms of that, I mean, I think, from some of our, like, I think these harsh environments of metal and glass, they definitely don't foster the same kind of microbial communities that you see in a natural environment. And that's one of the interesting questions, right, is that bad, is that good? And I think, we had, at least for a point in time, swung pretty far over to, we don't want microbes anywhere, right? We didn't want carpet because, that was building up dust and therefore germs. And, they were even selling antibacterial paints at a time, right? Because, gosh, why would we want bacteria on our walls? Like, no, they're there. But these are new questions, and the fundamental link here is, does spending 90% of your day, whether it's your apartment, your office building, in these types of structures, amongst these type of materials, is this bad for our health, and should we be,
Yeah, there's a
rolling
very
around in the
interesting
dirt? Yeah.
project going on right now under the auspices of the University of Chicago medical system and the Argonne National Laboratory, where they've actually taken a hospital that's been built from scratch. I mean, starting with a new construction site, the hospital microbiome project, and they have been tracking how the bacteria, the microbes that inhabit this site, change from when it's a hole in the ground, through when it's an active construction site, through when it's a completed building but as yet unoccupied by anyone except construction workers, and then as it becomes a full functioning hospital. And I mean, they're learning a great deal about how this built environment interacts with these microbes and vice versa. And an awful lot of what we thought we knew about hospital infection control turns out to be complete, if I can use the technical term, bullshit. They haven't finished yet. They got a couple of years to go, but these are open questions, and I think we have an open question behind you.
Hello. So this is a question from.
This is like our Delphic oracle, We turn to the woman in white for questions.
Okay, so this is a question from the amazing Robin Lloyd, and she asked, since most of us aren't doctors, how can we make good decisions about when to take antibiotics? And then she made an addendum saying that she just took some Pepto-Bismol, and how extensively has she changed her gut microbiome?
Yeah, Robin goes straight to it. Shall we flip a coin?
Okay, the antibiotics question is hard because obviously these drugs have done a huge amount of good for us. They've saved countless of lives from infectious diseases, and I think the message from the microbiome field isn't we should fear antibiotics, but we should use them judiciously, which is of course the exact same message that people have been saying with respect to drug-resistant infections, right? They are overused, it's a hard job, I feel. I don't feel I'm going to answer this question well, but I think it's a hard job for the individual to know how best to deploy them correctly. It feels like a matter for the medical profession, for agriculture. We need better tools. We need better diagnostic tools for working out what an infection is, for characterizing whether it is susceptible to this antibiotic or the other. But I feel like all of these things are happening now. Like there are big prizes in both the UK and the US specifically designed to develop the next generation of diagnostic tools. There is a huge amount of awareness being raised about the problem of overuse of antibiotics in a variety of different settings. And I think people are just getting a bit more savvy about this, and like the microbiome is part of that. But I feel like as individuals, it's a tough thing to ask. Like what do you, I think all you can do is just to not demonize them, but also not to crave them for everything. So don't like pester your doctor for antibiotics if you're only like mildly, if you've got a cold.
Yeah, I mean,
Yeah, so-
I was going to say, and not to call her out, but my own doctor, right before the exhibit opened, I had to go in for my checkup, and I broached this subject with her, and she said, well, yeah, I will sometimes give patients antibiotics because they're just going to get them anyway. And part of that is that we become very, very impatient, right? We're busy, we're losing work, and people forget that, an average cold can last 12 days, right? And so you think at day 5, this can't be a cold, I'm still sick. I'm going to the doctor knows you've got a cold, but you're like, you got to do something for me, or I'm switching doctors. So initially it was, I'll give you an antibiotic, it won't do any harm. And as Ed says, we've had this message in terms of drug resistance, but it still allowed us to be selfish, right? It's like, well, I'm going to feel better, screw you all, because it's going to evolve resistance after I've taken it. But now with microbiome playing a role, I think you can't necessarily play the selfish card so much. So, so we do have to be careful. I think, yeah, we're going to have a change in those practices. But to me, I think sometimes the individuals can come in more educated than the doctors. They have different incentives and different motivations. But Robin, in terms of the Pepto-Bismol, so I mean, I've bought 2 giant bottles of it because I'm going to take this every single day that I'm in the field, because bismuth is an agglutinizing thing, so it actually can help temper these microbiome changes. So it's this kind of a mild antibiotic without having a specific drug target. So you did something, but don't stress.
You have a question,
I'm
sir?
sorry you feel what.
Are you have a question, or are you?
Yeah.
Please.
So, in talking about the microbiome and the human microbiome, the microbiome as it spreads out into the world from us, each of us, and into different environments, we keep using the word microbe, and it's a wonderful word. But we also keep talking about the diversity of species and genre of microbes, and how do we balance the relative lack of diversity of terminology for microbes with the extreme diversity of microbes? We've got lots of words for cats because we're familiar with them, but we don't for, they're microbes that are wildly different from each other, and we call them microbes. We call them all microbes. So I was wondering if you could speak a little bit about that and how we can combat that and show people that they're
That's a very
different.
interesting question.
I mean, I was
Susan,
gonna say,
please.
this was actually one of the really serious challenges in developing this exhibit, because the museum exhibitions department is really good at dinosaur exhibits, and we had exhibits on whales and things. And so Rob DeSalle, who co-curated the exhibit with me, and we'd be like, they'd say, well, what's the species we can put on this? And we'd say, forget species. Well, what do we call this one? And it literally got to the extreme of the shop sending me pictures, not photographs, just drawings of microbes, and saying, could you tell me what species this is? And I was like, no, none of this means anything.
This is Bob.
This is
Bob
Bob.
the microbe.
Yeah. This is Harry.
Yeah.
And I think to me that has been one of the fundamental challenges of communicating about the microbiome, is that it is a whole different rubric, right? Part of it's because of horizontal gene transfer, right? So I love Claire Fraser used this example of, you'd have 2 E. coli who are less genetically similar than a human being and a banana. So they only share a small portion of their genome, but we would call them both E. coli based on some crude morphological characteristic we gave to them. So species is sort of meaningless. I think it's why studies that turn to metagenomics and look at collections of genes are actually probably the more powerful scientific way to answer questions about microbiome. As scientists, we don't care who's there so much as what they're doing, right? So I don't need to give every microbe that I might find on the subway or on my tongue or on my dog's paw a name. That there's a certain, there's a loveliness about taxonomy and describing that diversity, But in terms of answering big questions, naming them isn't getting us anywhere. It's really what genes they have and what are they doing and how do those things interact.
I think that's exactly right. And, you know, in any type of writing or communication, you work with where your audience is. And, you know, I've talked to friends who just about understand what a bacterium is, that know, like, there are microscopic things in the world around us. I talked to a friend of mine about this book, and she said, what are you writing about? I went, well, it's a book about the microbiome. And she went, what is that? Well, okay, well, so you know that there are like trillions of microbes that live with us. You went, wait, what? There are, so you're starting with like a point, you have a starting point where people don't even realize that these things exist. So does it matter that we're talking about bacteria and archaea and fungi and protists and the vast diversity? It's what the question is, what do you want to get across? And I think that Susan's exactly right. It's the themes you want to get across, much more so than the identities of the individual players. And talking about those identities is hard because these things don't have common names.
Right.
Like
Bob.
well, even like Wolbachia is sort of like you have the names that are like garbled mouthfuls to pronounce, and that even like trained microbiologists, like the number of people I've seen on stage messing up Bacteroid DTs and Bacteroides and Bacteroid Dales and a pox on whoever came up with all of. But actually, one thing I happily found while writing the book is that some of them do have common names, sort of. Like people who work on these things don't want to have to say the full name all the time, so you shorten them. So you get things like E. coli. Like E. coli is C. diff, exactly. B. frange, like, you know.
Yeah. Well,
someone suggested doing like a wrapper or microbe quiz.
It is true that the problems of scientific nomenclature are the problems of scientific nomenclature, but I do want to agree with our friend in his question that language is a form of cloaking, language is a form of avoidance behavior. There is a kind of intimacy here that I think generically people avoid. This is not science for the squeamish. We talk about fecal transplants, we're talking about crap. We're talking about mucus, we're talking about snot, we're talking about anuses, we're talking about, you name it. Yeah, and the reason your 4-year-old at your museum is directly in touch with it is because that 4-year-old probably still has not yet been totally enculturated to forget toilet training. In any event, while we have been sitting here in this room, I, like the rest of you, have shed about, give or take, 2 million microbes. You have released them onto your neighbors. You have let us go into the world because I'm now an us. I'm not a me anymore. I'm an us. And we have been really entertained, educated, and intrigued by the conversation that we have been listening to. And from the inner elbow to my diverse belly button to the joyful diversity of the microbes living between my teeth, I want to thank you for a wonderful conversation. And I ask you to join me.